So this is the first Nano webinar of the year. Happy New Year for those I haven't spoken to yet. My name is Lilian Krug. I am the scientific coordinator of POGO and of the NF-POGO Alumni Network for the Ocean. So the NF-POGO Alumni Network for the Ocean is an initiative started by POGO and the Nippon Foundation back in 2010, which objective is to keep track of the trainees after they conclude their training, but not only that, but maximize benefits from the training that we received, and provide further opportunities for networking and collaboration and research and experiences. So one of the major goals of the NF-POGO Alumni Network is to promote joint research activities among its members, and using this research towards the benefit of society. So since 2017, well, since between 2012 and 2017, we had regional research projects conducted in 5 different continents, involved more than 100 people on these research projects. They were very successful. And after they concluded, we considered that the natural way would have one big global research project, which is the NANO-DOAP. I usually introduce it, but this is not the case because Subrata will do it today for us. So talking about Subrata, he is the coordinator of the NANO-DOAP Global Research Project, but more importantly, he's an assistant professor in the Department of Oceanography from the Shahjalal University of Science and Technology in Bangladesh. His teaching and research areas include marine ecology and modeling of non-linear dynamics. Subrata's main research interest is to explore the impacts of environmental changes on community dynamics in the marine ecosystem. And in addition, he's also utilizing science-based findings for marine ecosystem management. He attended the NF-POGO Centre of Excellence in Observational Oceanography at Alfred Wegener Institute in Germany between 2013 and 2014, and is currently serving as the coordinator of this global project. So that's the subject of this webinar today. Subrata will present it for approximately 30 minutes, and then we open the floor for questions. So if you are comfortable with it, you can, first of all, using the reactions button, you can raise your hand, and then after having your name called, you can switch on your camera and microphone and answer the question, or put it in the chat, the question, and I can read it aloud. Another comment is that this seminar is being recorded, and it will go later on to YouTube, where we have a playlist of all Nano webinar series. If you're not comfortable with having your voice or face recorded and published, so please keep them off. Thank you very much, Subrata.
Yeah, thank you, Lika. Can you see my screen? Yeah. Yeah.
We can see the slides, yes.
Yeah. Yeah. So thank you, Lika, and hello everyone. So I have problem with sharing the screen, so I will put my presentation this way. So today I will talk about NANO-DOAP, which is an ocean monitoring initiative. So NANO-DOAP, Nano is NF-POGO Network for Alumni of the Ocean, and DOAP. So NANO-DOAP is a global project of NANO, which is the study on a global study of deoxygenation, ocean acidification, and productivity at selected sites from Africa, Asia, and Latin America. So as I said, the project is on ocean monitoring. So why ocean monitoring is important? So we all know the ocean covers around 71% of the Earth's surface, and land covers only 29%. So we can say that it is well known that the Earth is a watery planet. And then half of the global net annual photosynthesis occurs in the ocean. That means about half of the total primary production of the Earth comes from the ocean. And in addition to this, ocean absorbs about 31% of the carbon dioxide. That means ocean is absorbing heat. So ocean acts as a buffer against the temperature rise. As I said, the most of the primary production comes from the ocean, and this primary productivity is required for sustaining the fisheries. And ocean contributes about 81.5 million tons of global fisheries annually. And ocean is also source of energy, transport, and recreation. And it is also estimated that ocean is contributing about 500 billion USD to the world's economy. And the ocean-based economic activities, which is widely known as blue economy, and this blue economy is offering more than 31 million direct jobs to the people. So ocean is very much important for us. But with the increasing number of population and with the increase of fossil fuel burning and greenhouse gas emission, the environmental conditions of ocean are changing. And these changing are causing a serious threats to the ocean ecosystem. For example, here you can see the atmospheric carbon dioxide is rising over the period, and this is positively related to the rise in sea surface temperature. So carbon dioxide is rising, sea surface temperature is also rising. As a result, what is happening? The pH is declining. So we all know if pH declines, the water will be acidic. And as the temperature is rising, it also changes the oxygen concentration in the oceanic ecosystem, because rise in temperature reduce the solubility of dissolved oxygen in the ocean ecosystem. And long-term data measured from different parts of the ocean showed there is a deoxygenation in the ocean ecosystem. And here you can see the spatial distribution of oxygen concentration. And in some areas, oxygen concentration is low. So we can see the deoxygenation is happening. So if oxygen declines, it increases the respiration, and it also reduces the primary productivity. And here you can see the decrease in global primary production in the ocean. So we can now see that environmental condition is changing, and this change is altering the ecosystem health of ocean. So if we want a sustainable ocean, we need to monitor all these parameters. So Nippon Foundation and Partnership for Observation of Global Ocean believe that joint initiative can achieve a sustainable ocean monitoring activities. So in line with that belief, NANO started the regional research project between 2013 and 2017, which was funded by Nippon Foundation and POGO. And these projects, regional projects, are conducted by NANO members. So the NANO members, I would like to say the scholars who participated in different training programs of POGO, they are the member of NANO. So these projects are of these members usually participated in these projects, and in regional projects, a lot of issues are conducted. For example, in the regional studies, study on harmful algal blooms, tides, currents, erosion, and exotic species invasion. All these topics are studied. And this regional research project inspired NANO to start another great initiative, which is known as NANO Global project. In short form, we call it NANO-DOAP, which is a global study of deoxygenation, ocean acidification, and productivity at selected sites. So this project is in operation in countries from Asia, Africa, and Latin America. And the overall objectives of these projects are support in situ measurement of selected essential oceanographic variables, and then compile the data set. And this project also provide the capacity building opportunities. For example, this webinar is also a capacity building option. So in this type of webinar, we always organize from the project. And this webinar, we invite people, especially the NANO members, to share their research with others so that it can develop the capacity of the project participants. And these projects, these webinars are open to all. So non-NANO members are also able to join in this webinar series. And we also initiate and promote outreach and citizen science activities from this project. So currently, we have 30 sampling sites from 18 countries. So here you can see distribution of our sampling sites from different countries. So in some countries we have multiple sampling sites, and in some countries we have only one sampling site. So representatives of these sampling sites are NANO members, and they are responsible to do sampling. So most of the sampling site representatives do sampling on temperature, salinity, pH, dissolved oxygen, chlorophyll A. So these are 5 mandatory variables that we all need to measure in this project. However, few participants also measure nutrients, plankton, total alkalinity, turbidity, sedum. And all participants measure this data from the surface water. However, few sampling sites also measure from different depths, and depths can vary up to 80 meter or 100 meter. Most of these sampling sites do monthly sampling, and few sampling sites also do bi-monthly sampling. So since we started this project from 2018, so we have data from 2018 to until now. So it's almost 3 years. So 3 years we have data. But now it's time to do some analysis. Especially we need to do the interstation comparison. But when we wanted to do this, we faced some problems because the institutes who are involved with this project, they have different types of capacities, and they have different types of instruments. So for the same variable, they use different types of instruments. For example, some sampling site use CTD to measure temperature, salinity, and chlorophyll, and some sampling sites use water multimeter to measure the temperature, salinity, and some sampling site is using fluorometer to measure the chlorophyll. And so there is a diversity in sampling instruments. The main problem with this diverse instrument is that their accuracy and precision vary significantly. So this creates a problem when we want to do the interstation comparison of this data. Therefore, in this year, I mean, during 2021 to 2022, one of the major objectives of this project was to develop a method for interstation comparison of data. So one way to deal with this problem of multiple instruments from different sampling sites is to make paired measurements using both instruments to see how well they agree or don't. But this is very difficult. I would say this is rarely possible. And then next best step is to use the precision and accuracy information of the instrument from different sampling sites. So for that, first we compiled all the data in one database, and then we choose 14 sampling sites because from 2018 to 2020, about 14 sampling sites monitored measured the data continuously. And then, as I said, the depth varied. So when we took 14 sampling sites, then we found around 36,000 sampling points. And since the depth of measurement varied from sampling site to sampling site, so we considered only first 5 meters, and then we considered only temperature, salinity, pH, dissolved oxygen, and chlorophyll. So we considered only 5 variables and data for first 5 meters. So then we had the data for about 6,000 data points. So first it was like 36,000, and then it reduced after sorting to 6,000 data points. And then we collected the accuracy and precision information of instruments from our project participants. As it is a bit complex due to the variation of instruments, accuracy and precision, we had to do some statistical analysis. So for that, we tried to review all the methods for the standardization of the data. And then we found 3 methods that already worked for the standardization of the data. So I will explain these a bit more. So Panasa et al. 2018, they developed a method for data standardization. So in their study, they standardized the data from 5 hydrographic cruises in the Antarctic intermediate water. So these data sets are collected during 1990 to 2014. So here you can see the locations of 5 hydrographic cruises. So this is the transect for 1990, and then this is the transect for 1992, and then this is the transect for 2008. So here you can see there is some variation in sampling locations. So this might give some error in the analysis. So to minimize this error, Panasa et al. Developed a method to standardize their data. So what they did, they actually developed some adjustment factors for their analysis. So for that, they first extracted all the parameters from all the cruises, and then they regressed each parameter against temperature. So the reason of regression of each parameter against temperature was that because temperature they found as the least variable parameter among these 5 cruises. So they regressed all parameters against the temperature, and then they obtained the regression. Then they obtained the required adjustment for each cruise by taking the ratio between the mean intercept of all cruise and the intercept of each individual cruise. So let me visualize this. So as I said, first they extracted all the data, and then they took temperature as the standard variable, and then they they extracted all the data for temperature and all the data for salinity, and then they did the regression analysis, and then they estimated the intercept, and then they regressed temperature for cruise 1 with temperature salinity for cruise 1, and there they got another intercept. Let's say intercept y for cruise 1, and again they did a regression for temperature data from cruise 2 and salinity data from cruise 2. And there they got another intercept, say Z. So then they estimated the adjustment factor for cruise 1 for salinity is equal to this intercept, that is X, and then divided by this intercept. And next, when they estimated the adjustment factor for cruise 2, what they did the same thing, intercept for all cruise divided by intercept for cruise 2. So this is the adjustment factor for salinity, and similar way they estimated the adjustment factor for pH, dissolved oxygen, and also for nutrients. And similar method is also applied by Sarker et al. And this method was applied to the for the same type of study in the Bay of Bengal. So here you can see these are the transits from different years. So there is some gaps. There are some these may make some error in the analysis. So they also use the same method for standardization of the data. And then we also used another method developed by Sarker et al., which was used by comparison of the phytoplankton data collected by different person at different time. So for this, Sarker et al., in this study, they used a data set of phytoplankton from Helgoland Roads time series station. So this time series station has the data since 1962, and the data set has about total 365 species. So they had the data for 365 species from 1962 to 2015, but all the species data are not monitored regularly for this whole time period. So they had only 26 species of phytoplankton data, which was monitored continuously from 1962 to 2015. So if we want to see the long-term trend in phytoplankton diversity or phytoplankton community structure, we can only use these 26 species. But a important question arises: if we use 26 species out of 365 species, is it like logical? Because use of 26 species out of 365 species is short of thinning. So, but there are no other way to do this, because since only these 26 species are monitored continuously. So one option is to see if these 26 species represents the entire community of 365 species, then we can use this 26 species for our analysis. So now we need to check if they really represent or not. So what we can see from 1990 to 2015, all the species are monitored. So for the test, they used the data of 365 species from 1990 to 2015, and then they randomly choose some replicates of species. Like they first choose 30 species randomly from 1990 to 2015, and then they choose 40 species, and then they choose 50 species. Like this way, they prepared the 10 randomly chosen phytoplankton community. And then they estimated the Shannon index for each of the replicated community. And then they estimated the ratio of Shannon diversity index for 365 species for this 1990 to 2015 time period, and the Shannon diversity index for each of the replicates. So, and then what they found that even if they increase the species number, the ratio between 365 species and the replicated community, the ratio is near to 1 or within the plus minus 10% of 1. So if the ratio is 1 or near to within the limit of plus minus 10% of 1, then we can use the 26 species as the representative of the entire community. So now going back to our story. So when we discussed about the paper of Panasa et al., they had the problem of sampling from different locations. In our case, we have problem with these different sampling instruments. So if we take their method, in our case, what we have to do, first we have to choose a variable. Like Panasa et al. Regressed all parameters against temperature. So we also need to find out a variable which is least variable in terms of accuracy and precision of the instruments. And this variation should be statistically insignificant. So to find out a variable in which is less variable in terms of accuracy and precision of instruments, we had to do some statistical analysis. So first we collected all the information of accuracy and precision of the instruments, and then all possible values for instruments with the accuracy and precision range are generated. So we generated these values with Monte Carlo simulation. So for example, with an instrument, we measured the temperature from a sampling site for a certain time period is 25 degrees Celsius, and the accuracy of the instrument was plus minus 1 degree Celsius, and precision is like 0.01 degree Celsius. So all the possible values could be between 24 to 26 degrees Celsius with the precision of 0.01. So what we did, we simulated all the temperature, all the possible temperature with multi-color simulation between this range, that is 24 to 26. And then like what Sarker et al. Did for their phytoplankton study, we estimated the ratio of simulated temperature and the actual temperature. And then if the ratio is 1 or within plus minus 10%, then we can use that particular variable as standard variable. So we did this for all stations or sampling stations for all variables. And then we found that temperature is the least variable. So here you can see temperature, the ratio is within the plus minus 10% limit for all sampling stations. So we took temperature as the standard variable. And then we estimated the adjustment factors like Panasa et al. Did. We compiled all the temperature data from all stations, and then we extracted salinity data for all stations. Then we estimated the intercept, and then we separated the data station wise. So this is like temperature and salinity data for station 1, and then we estimated the intercept y, and then we did for station 2, station 3, station 4, like all stations. And then we calculated the adjustment factor for station 1 for salinity is like x by y, and adjustment factor for station 2 salinity is x by z. And we did this for all stations that we had, and we did this for all variables, especially the 5 essential oceanographic variables that we measured. And then we did the seasonality analysis, and we also did some analysis about the spatial trend. And we also aim to do the causality analysis because my presentation is mainly focused on the methodology that we use to standardize the data. So we did not explore so much the standardized data yet, but I will show you some preliminary results from this analysis. So here you can see variability in surface temperature. So we divided the sampling sets into different regions like equatorial region, subtropical region, and tropical region. So you all see it's also trivial that there is a seasonality in the data and the spatial variability in data, and near the equator temperature is higher, And it is important to see that near the equator, the amplitude of seasonal variation is low compared to other area. So, and then here you can see the variation of temperature, seasonal variation of temperature in subtropical coast, subtropical estuary, tropical estuary, and tropical coast. And this is also the same. We have the seasonality in the data, and but amplitude varies from different locations. And then this is the spatial variability of sea surface temperature. So this is the latitudinal variation of sea surface temperature, and we can see the substantial latitudinal variation in sea surface temperature, and higher temperature is observed in the near the equator. And if we compare this pattern with this satellite derived data, we can also see the similar pattern in the data set. But for salinity, when we analyze salinity, we got some trend, a special trend. But here you can see that these data points are they showed very low salinity. So this is because our sampling sites are a combination of coastal site and estuarian site, and some sites are influenced by upwelling or strong current. So these are the reason for the variation of the salinity. We still need to think about how we have to we can incorporate these characteristics of sampling sites with this analysis. Maybe we can we will do this later. And then this is the result from dissolved oxygen. Here you can see high dissolved oxygen we observed in the subtropical coast, and comparatively low dissolved oxygen was observed in the equatorial area. This might be related to one of the reason could be like high temperature, which reduce the solubility of the oxygen in the seawater. And then this is the latitudinal variation of dissolved oxygen. Here you can see in the equatorial area, we have the low dissolved oxygen concentration, and we tried to compare this trend with the NANO-DOAP database. So we also found similar types of pattern in the dissolved oxygen concentration. And then the pH varied between 7.8 to 8.2 in our study areas, and high pH we observed near the equator. So, and then we tried to see if the different areas have the unique characteristics in terms of the environmental condition. So we performed an MDS analysis by taking the 3 regions data, equatorial, subtropical, and tropical. So we found that each of the regions have almost unique environmental characteristics. Here you can see all these sampling sites have different clusters, but we have also seen some overlapping in the data. So these are some preliminary analysis from the project, and chlorophyll data we still need to analyze, and we still need to improve our statistical analysis. And so from this preliminary analysis, we have some conclusions. We observed the strong spatial variability in your essential oceanographic variables across different sampling sites. And as we see the temperature rise and the drop, and we also seen the pH condition. So if temperature is temperature shows the rising trend, and pH goes down and DO also goes down, this will disrupt the ecological dynamics of the ocean. So one of our project objective is monitoring, and we also suggest that continuous monitoring of global ocean is required. And for that, of course, we need joint efforts, because single efforts cannot achieve this goal. And one of the important learning from this project is homogeneity of instruments for this comparison of the data. The problem arises for this interstation comparison for the variation of precision and accuracy of the instruments. So homogeneity of instruments is required. This is essential. We know it is very difficult to achieve, but this can be done through developing low-cost instruments, which is another objective of the POGO. So if we can use low-cost instruments and we have joint efforts, then it will be possible to monitor our ocean. So the project will also continue for the next year, and in the next year we will continue our monitoring of these oceanographic variables in different sampling sites. And we also aim to expand our network through joining of new nano members in the coming year. And we will continue our data analysis, data compilation, and preparation of our database. And we will also continue our outreach, citizen science, and capacity building activities. And of course, we will improve our statistical analysis for the interstation comparison. And I would also like to ask all of you to give you thought and your suggestion to improve our analysis. And even if we can do that next year, we'll try to put all these findings in a scientific paper. Thank you.
Thank you very much, Sub. And it's very nice to see this progress in the project. So I'm opening for questions. Again, this is being recorded and later uploaded to YouTube. So if you're not comfortable having your voice or image recorded, you're free to welcome to type your question in the chat and I can read it to Subrata. Lamona, yes?
Yes. Hello. Hi. Nice to meet you again.
Nice to see you.
I'm sorry I cannot open my camera now. It's late and I'm not ready for camera. Yeah. And Subrata, very nice presentation. I would like to ask about the one you make comparison for the intercept for each regression for each station. Yeah, and then so we have a adjustment factor.
Yeah.
Yeah, and then so what do we do with this adjustment factor? We just multiply our data to the adjustment factor and then use it for the further analysis.
Yeah. So like when you develop the adjustment factors like for salinity, so let's say the adjustment the ratio is like 0.01. So you just multiply the for each station you will have one adjustment factor. So just multiply the data set that was given by the participant by this adjustment factor.
Ah, okay. Yeah, so then we can use for the further analysis. Yeah, but first we need to multiply each data with the adjustment factor.
Yeah,
okay,
each
okay.
point.
Yeah, so the adjustment factor was the first the all station intercept divided by the one station intercept. Yeah.
Yes, individual station intercept.
Yeah. Okay. Thank you very much.
Thank you, Lana.
Can I include a question on this? Saying my concern is SOA's and yours papers were doing these adjustments for cruises and different data sets that were sampled pretty much in the same region. So it's kind of a twofold question. The first part is that how confident are we of using this strategy of adjustments between data sets that are sampled in such different conditions? So, you know, we have stations in the open ocean and we have stations within estuaries. Do you think that this will have an impact?
Yeah, for the, I think it will not have impact. It will not have an impact because we are expecting the variability in the sampling size, and we are expecting the problem with this the variability of measured values due to the instruments. So this is our main concern, and and what we did also, we tried to measure the data with the different instruments at the same time from Bangladesh sampling sites. Sorry. And then we applied this method to see if this method really improved the results. And then we found that if we apply this method, it really improves the result. So if that is one of the concerns, then we can also test this idea in 1 or 2 more sampling sites, and then we can see if we can do this in one estuarine area and one in the open ocean area. So
The
and then we
adjustment
can apply
factor.
this method.
Yes,
Yeah,
I think
we
that
can
maybe
see
that. Yeah, sorry, please continue.
yeah, no.
Yeah,
So
I
it will
think
be
that maybe this is something to take into consideration, because some stations can be quite different, and this is also something that we could use, for instance, on the salinity plot. That, you know, the latitudinal comparison, it's not really showing a good way.
Yeah,
a good way to measure would maybe distance from the coast, or something like this could be also integrated. And then the second part of my question is, and I'm not sure if I got this right, but the adjustment factor takes into consideration linear relationship between temperature and the other variables. But do they really have a linear relationship? For instance, chlorophyll and temperature, is this really linear?
For chlorophyll, it's a bit messy. So we
Yeah.
haven't included the chlorophyll in the analysis yet. So we need to think about that, because chlorophyll is really messy.
Okay, thank you. But yeah, it's really nice seeing coming along. And on the conclusions, we are just starting. We have a little bit over 4 or 5 years of data set, so it's really difficult for us to talk about trends right now. But you know, this is the starting of something really nice. We started with 9 stations or something like this, and we are currently on 31. So I see a lot of hope in our project. Just before I give the floor to Lamona, I want to read. So Mohammed has a question on the chat. Hello, colleagues. Subrata, maybe you can share the paper used for the regression of data with temperature. We can read further to understand the concept. And I agree with Lika, different systems would have different primary factors driving the variations. Ignore the last word. Yeah.
Okay, I will share this. Yeah.
We do. I believe we do have it cited in the quarter report that was circulated to members, Mohammed.
Yeah.
You can find the reference in the report.
And
but
it's open access.
It's open access, yes. Lamona?
Yeah, thank you. Okay. Then I think of something that we have already this adjustment factor, but it's based on the linear regression between the salinity, the temperature and the salinity. Yeah. And what do we do with the, for example, the pH, the dissolved oxygen? Of course, we do not use the adjustment factors from the TS relation. Yeah, we need to make their own adjustment factor. Do you think?
Yeah.
So, for example, between the temperature to DO, to the dissolved oxygen, and then or the temperature to the pH, probably.
Yeah.
And then, yeah, you said that the chlorophyll was messy, and
And one just information, and in this we have a very large data gap for chlorophyll. So in many stations, chlorophyll data we do not have.
Yeah.
Okay. Yeah. So then how do we determine, for example, for adjustment factor, TS is really, it's a very general relation. I mean, we use TS diagrams, etc. But for example, if we'd like to analyze for the DO or pH, for example, which other parameters should we pair up to? For example, pH with temperature or better with salinity. And how do we choose one?
Okay, at this moment we are using the temperature because temperature we found the least variable in terms of accuracy and precision. So this is like we are making some constant values that will just improve the strategy to comparison of the data.
Okay.
So in all cases, at this moment, we are considering the temperature.
Yeah, so basically there's a lot of variation happening, Lamona, because, you know, besides the fact that there are different, that each sampling station, it has its own characteristics. Each one has their different sets of instrumentation, sampling data. They also cover different depths, different frequency of sampling. They are joining the project in different periods as well. So there's a lot of different, and Subrata and Samuel are doing a great work trying to find the commonalities and excluding, you know, variability to try and make a nice comparison. And it's surprising that we have already so many data. I mean, if we consider surface data for all this, it's 6,000 data points, you said, Subrata?
Yeah.
For temperature? No, for all the 5 required.
For all, yeah, for all 5 parameters.
Yeah. So that's why it's nice that the representatives of the stations keep up with their regular sampling, particularly for these 0 to 5 meters data depth.
Yeah.
Okay.
Another important issue for chlorophyll is many of the participants use spectrophotometer for chlorophyll measurement. So in that case, we do not have actually information of accuracy and precision.
But I use fluorometer.
Yeah, you use fluorometer, yeah?
Yeah.
Okay, another comment in the chat, it's from Hussam. Hussam, hi. Thanks, Subrata et al. Nice work, great presentation. Thank you, Hussam.
Hey guys.
Hi.
Thanks. Hey there. Thanks,
This is
Subrata.
the former coordinator of NANO-DOAP, guys.
Yeah, I can see. I'm really happy to see how far you gone, guys. Like with Subrata, great work. Thanks a lot. You know, I've been listening carefully, and yeah, thanks a lot.
Yeah, this is his baby. It was
yeah.
Hussam who put a lot of work on bringing the people together and discussing, strategizing.
Yeah, and now
sampling.
I'm so happy to see that. So it's in a really good hand, and Subrata is going further, especially in the data analysis and making sense out of the database. That's really great. I'm really happy to see that.
Great, thank you, Hussam.
You're welcome.
We are very lucky to have both of you leading this. I think if there is there any other questions? We are reaching the 1 hour line. Esoe, hi, please.
Yes. Hello.
Hello.
Hello. How are you doing?
Very good, thank you. How are you? Happy New Year.
Yeah, Happy New Year to you and all the members of NANO-DOAP. And first of all, I would like to thank Subrata for the nice talk. And I don't have at the moment many comments, but this is probably regarding the adjustments that Subrata referred to our paper and all this. And maybe, I mean, Lika already highlights about this. Subrata, you already find out that temperature and prove that is the most least variable that has less precision. I can say that. And now the best things will be that you consider different area, like we are sampling in different coastal zones that are sometimes affected by other factor. I don't know, I can say we are in the range tropical water here, and somewhere some other people are in some type of the water. Then you can make like regional correction, and then you can, because you already do like Monte Carlos and all this approach to find out the temperature range. Then you can maybe try to find out or yeah, like I'm telling you, different region, and then try to apply this correction factor, and then we can see if we still the correction factor makes sense or not. But that's my suggestion. But we can, I will discuss this with Subrata later. Thank you very much.
Thank you.
Yeah.
You mean instead of using an intercept from all stations, have different intercepts for stations that are classified together, like
exactly together. Exactly.
Yeah, that's a nice idea. We can try that.
We can see. Yeah.
Yes, it would be very nice to have not only you, SOA, for those who are not aware, SOA is the NANO-DOAP member from Togo, and the author of the paper we are using for the methodological approach. So it would be really nice to have your inputs as well, SOA, not only yours, but Muhammad and Lamona and everyone. Hi, good to see you.
Hi,
I'm afraid.
SOA. Nice to see you again.
I'm
Yes,
afraid
nice
to
see
switch
you,
on
Lamona.
my camera because
no problem. But for me, I'm not afraid too much. But I'm very happy to see everyone for a long time ago. Lamona.
Yeah. Hi.
Let's give
It's
our
been
greeting.
5 years.
Yes, of course.
You guys have your
we
little
have the
club,
beer and all this now. Thank you.
Yeah.
Thank you. So nice.
And next time I will make sure I open my camera.
Yeah, no problem. Just this is for Lamona so that she can see me again for a long time ago.
Very nice. Thank you. We used to work at the same building in Bremerhaven. Yeah.
Yes, of course. Of course. Thank you,
You
very
look
much.
very good. You look very good.
Thank you.
Yes. Okay. So we will keep in touch, and it would be really nice to have this little, a little team within NANODOAP to discuss this and support Subrata on the correction of data for the interstation analysis. I think that we have to call the day, but I want to thank Subrata and everyone really who attended this webinar and give their inputs and just watched and supported our project. I thank everybody once again. This webinar will be soon uploaded to YouTube, so you can watch again later on. For Nano members, both presentation, the presentation PDF will also be available on the NANODOAP website. And it's at www.nf-pogo-alumni.org. And we will be available for questions here and in social media as well. Okay? Okay. Thank you, everybody. Have a nice weekend.
Yeah, have a nice weekend, everyone. Bye.